Hi, Kyle. So, could you please introduce yourself so our listeners know who you are?
Sure, great. It's great to be with the Longevity Circle, and thank you so much for this invitation. My name is Kyle Citrullo. I am the president and founder of the International Perinatal Stem Cell Society, which is a nonprofit organization, the 501(c)(3) that I started in 2013. And the goal of that organization is to bring affordable and efficacious stem cell therapies to the United States. Obviously, we've been working for many years on that, but we're really seeing some breakthroughs, and there's new state laws, and there's a lot of really exciting things happening in the space. But what I'm here to talk to you about today is this new revolutionary subset of stem cells called the Muse stem cell, and I'm really
Yeah.
looking forward to dropping in with you on this because it's so exciting.
It is very interesting because I'm personally I didn't really hear a lot about that, so I'm just like learning as I go like in this industry. So, yes, we will be like talking about like the new era like in longevity and like about the Muse stem cells. And as I understood, the Muse stem cells, it's different from the regular, like what we hear about the stem cells, correct? So I will just let you to explain like what is this, like why is different, what the benefits, and we'll let you to do the presentation.
Exactly, cool. Yeah, I'd love to drop in. So I'll share my screen. So what we're going to be talking about is the uh, the perinatal stem cells, and that has to do with around the time of birth. So it's really the birth tissue, and it's the stem cells that happen in this miracle of childbirth, where this baby develops in a woman's womb. And what we're really talking about are the stem cells that are in this umbilical cord. And these cells are just amazing stem cells. And these are the stem cells that we hear about when people go to Tijuana or Costa Rica and get the expanded umbilical cord mesenchymal stem cells. And why I call those like the Joe Rogan stem cells, where the celebrities are traveling offshore. And the reason they're traveling offshore is because technically when you culture these cells and grow them, that's considered a drug in the United States in at this current time. But that's being those restrictions are being lifted right as we speak. I mean, they've changed in Florida, Nevada, Washington State, and Utah, and it's really becoming like a cannabis states' rights versus federal rights play on stem cells, and many other states are coming as well. And additionally, there's a new law that was passed in Montana uh, that made changes or amendments to a federal law, the Right to Try law, which I'll get into in this presentation as well, because it's really going to revolutionize this field because it's going to enable doctors and to offer these treatments in the United States and for patients to access the highest medical care and get stem cells, which is going to be really amazing. But this period of time of birth is an incredible period where these stem cells are incredibly potent and they're able to be harvested without any ethical issues at the time of birth. So that's additionally, these stem cells are also closer to the embryonic stem cells in the time that they're developed. So you get stem cells from multiple sources, with bone marrow and fat tissue being two of the major cell sources, but those cells are not as potent and as versatile as the perinatal stem cells. And if you go back further to the embryonic stem cells, those stem cells are the George Bush controversy, where you had to kill a blastocyst to collect those stem cells. And those stem cells are actually too potent. And as soon as you
When you
take them
have
out
to
of this,
kill, sorry, who?
they're too potent. So in the sense that when you take them out of this pristine womb environment and put them into a laboratory and direct them to go to different tissues, they form tumors. So this
Oh.
perinatal source is kind of this perfect sweet spot where they have the potency in some of these embryonic markers, but they don't have any of the tumorigenicity. So there's been over 30,000 peer-reviewed studies studying the umbilical cord mesenchymal stem cell, and the safety profile is just credible. And they also have advantages uh, that they actually home to areas of inflammation naturally. So for any
But
injury or any
yeah,
area that is inflamed. Go ahead.
I have a question, but if there are like so many studies have been done and it's super safe, why FDA still didn't, like clear like this treatment?
I mean, that's one of the biggest problems with the field. I mean, the mission statement from my nonprofit, which we started in 2013, was to allow access in the United States. But the FDA has just been very slow, and also the mechanism to get a stem cell process forward is the same as developing a small molecule drug. You have to go through phase three, phase one, phase two, and phase three clinical trials, and at the end of the day, there's really nothing to patent on the stem cell. So you can't patent a biologic, naturally occurring cell. So unlike a small molecule, you can patent that. So what happens is there's actually, there's a lot of conspiracy of whether pharma doesn't want these stem cell products to be out because they'll actually cure us. But what really happens is there's actually a period in the field of regenerative medicine, particularly in cellular therapy, called the Valley of Death. Now, that's not a good moniker to have for any business, but basically, the Valley of Death is when a company has had a successful phase two trial that shows that their stem cell product works, but they're unable to raise the money to go to the phase three and actually finish the product. And there's a number of reasons. Mostly is because if I successfully did that and I came out with a blockbuster, you could follow right behind me. And then the competition is, the field is cut in half. Whereas you have 17 years exclusivity if you're developing a small molecule. So that tends to be where they put their money, because then they can have an exclusivity domain. And then the other thing is just the way that the FDA reviews these trials take a long time. And often what happens is you'll meet with one FDA reviewer and they'll tell you to go do all this extra work. And then you'll go back with that extra work and there'll be another reviewer there who just needs to get back up to speed on your drug development and will not understand it and have to send you back to do more development. There's lots of examples where that has happened, but mostly the state laws are changing that, because the state laws are allowing this to happen with relatively limited supervision, and that's one of the areas that we need to watch. But basically, the access is going to be available, and it was already available in multiple states, particularly Florida and Utah and Nevada, where these expanded umbilical cord cells are legal. Where actually it says non-FDA approved stem cell therapies are legal. But what I
Okay.
want to really talk about the Muse cell today.
Yeah.
So I'm going to just go forward a little bit here. This is my nonprofit focuses on providing stem cell therapies to veterans at this point, and we've partnered with the Sabot Foundation as well to do that. So every month we find a cohort of veterans and we get companies to donate stem cell therapies or product, and then we work with different doctors to deliver it. But I really want to get into the Muse cell. So the Muse cell, the stem cell therapy that is the most effective is the expanded umbilical cord stem cells. As I mentioned before, they're this sweet spot of development where they're safe, but they're also incredibly potent, and they don't need to match either. So my mesenchymal stem cells can match with anyone's stem cells as well, and vice versa. So it's this universal stem cell. And this is what you think of where all the MMA fighters are traveling to Colombia or Tijuana to get treatments, because there's some really excellent centers that do that and offer this therapy. But this therapy is nothing out of ordinary that couldn't be offered in any American hospital or any real clinic. As you know, it's not basic, but it's not difficult either. If you have a PhD in the lab, you can really make some really great stem cell products. And what the Muse cell in particular, and we call it this Muse cell because it has these markers of embryonic stem cells. This is a paper that I wrote with my previous company, Oxocell Laboratories, in 2011, where we identified that the mesenchymal properties, exhibit properties that are attributed to embryonic stem cells. So it's these high potency markers, particularly SSEA4 and SSEA3, which are embryonic markers that have to do with the ability for the cells to become all three germ layers, which means they can become muscle, nerve cell, bone, you name it, like any type of tissue cartilage tissue, pancreatic tissue, liver tissue, nerve tissue in the brain. So this is the type of area that we want to focus on for regenerative medicine. And the Muse cell contain these embryonic markers uh, that even the other umbilical cord mesenchymal stem cells do not. So although I talked about the sweet spot being the embryonic, in between the adult stem cells and the embryonic stem cells where the perinatal stem cells reside, within the mesenchymal stem cell population, about 1% of those cells are these supercells. And these supercells are defined by their expression of these two markers. And the first being SSAA3 positive, and the second is the HLA-G. And HLA-G is an incredible marker because HLA stands for human lymphocyte antigen. And what HLA is, when you've heard of a bone marrow donor, where you have chemotherapy and radiation, you need to rebuild your immune system. You have to do a stem cell transplant with the type stem cell hematopoietic stem cells. And you match that. That needs to be highly matched, and you match on an HLA level. So HLA-G is a marker that's expressed in the womb and goes with all the stem cells that allows a baby to develop in the mother's belly without being rejected as a foreign substance. Because it's obviously foreign cells. But this HLA-G, it cloaks those cells so the mother's body recognizes them as their own. So the Muse cell is the subpopulation of the umbilical cord mesenchymal stem cells that express HLA-G at 90%. So what that means is that it doesn't get rejected by the body. So if you have 100 of these cells, one of those cells is going to be the supercell expressing this HLA-G at 90%, whereas the other cells express it at 20%. And then also the SSAA3 positive, along with some other embryonic stem cell markers as well. So it's a super potent cell that doesn't get rejected in the body. So a regular MSC treatment will include about 1 to 2% of these Muse cells anyway. So anyone that's got a stem cell transplant has already received some Muse cells. So the safety profile could be extended to the embryonic stem cells because these Muse cells to the mesenchymal stem cell transplants that happen all the time offshore and now in the United States. And they also will, the way that a regular mesenchymal stem cell will work is it will stay in your body for 3 to 4 weeks. It will home to areas of inflammation, and it will go there and release growth factors, exosomes, cytokines, and other signaling mechanism to tell your body to change from an inflamed, like wounded state to become a healing state. And oftentimes, chronic pain or chronic injuries, if you think of like a diabetic ulcer, like a wound on someone's leg that just will not close. And yes.
Yeah, I have a question. So basically, like if I understood everything correct, so the Muse stem cell is the one that is coming from like from the baby, right? So umbilical cord.
Cord. Yeah.
And so it's better than we're just talking about just the regular stem cells, which we can take from the blood, right? From the fat, and from the bone marrow, correct?
Correct. So that's half correct.
Yeah.
So the
And
umbilical cord stem
yeah.
cells have advantages over the bone marrow and the adipose to begin with.
Okay. And then as we know, like from the medical history, sometimes when we try to inject, like something that doesn't belong to us, like doesn't belong to like our body, like not our DNA, so the body is trying to fight it or like doesn't recognize it. And so like in order to do this, like inject, like someone else, like Muse cells, like how does the body like affects, like what's the reason, for example, for the body not to take it as a something foreign and like not fight like against that?
Yeah. So there's different type of stem cells. So from the umbilical cord as well, there's umbilical cord blood, and that's a hematopoietic stem cell, which recreates all the blood cells. So the white blood cells, for instance. So after chemotherapy, that's when you get a stem cell transplant from cord blood or bone marrow, and that reconstitutes the immune system. That cell needs to match on a high degree of HLA specificity. Otherwise, it will be rejected and cause graft versus host disease. And sometimes it does as well. And the body really fights it. And that's like, that causes 5% of all deaths in cancer treatments, is the post reaction to the stem cell treatment. The mesenchymal stem cell does not need to match on that level. So it's a universal stem cell, but your body will still see it as foreign after a while because it expresses only 20% of this HLA-G. So after about 3 to 4 weeks, your body will remove those cells. So they'll no longer be circulating in your body. But what they have done already is they've changed the niche environment of the injury. So they've gone in and they've signaled to the body to start healing, but it won't actually make new cells. So if you do like a direct injection to a knee and you're looking to make new cartilage, for instance, those cells, those mesenchymal stem cells, even though they're stem cells, are not going to integrate into the knee and become new cartilage cells. However, that 1% fraction that are the Muse cells will. So the Muse cells will not only stay in the body 6 months or more, they'll also directly integrate into the tissue. So there's a stroke paper, for instance, where they bioluminesced the Muse cell. They did an IV of 15 million Muse cells, and it showed that it homed to areas of injury. So it went to the brain where the stroke injury was. It showed that there were still bioluminescent cells available, in the brain at the site of injury 6 months later. And they also showed that they became nerve cells that directly integrated into that patient's brain.
Mm.
And that is what the
Yeah.
Muse brings to the table, which is incredibly significant because it can become new cartilage, it can become new bone directly. And
Yeah.
that's the huge improvement of the Muse cell versus even the regular MSC. There's a slide I'm going to jump ahead to because so this is a great example of this slide. This here is a knee injury, and you can see
Mhm.
here the injury here with no stem cells. This is the 99% of the cells, the regular MSCs, the ones that are currently
Mm.
being offered in the world. In the Joe Rogan type stem cell where people go travel offshore, you get this level of healing. Now, people have excellent results at this level. They say, "Oh, I got stem cells offshore and I'm so improved." That's this level. That's the 99% of the cells. When you take the 1% and grow them up, this is the type of healing that you get. It's a full closure of the wound. And same
Mm-hmm.
with additional studies. There's a diabetic skin ulcer wound. The what I was just talking about, the diabetic wounds that won't close. With the Muse cell, they had 100% wound closure. Um, in,
Mm-hmm.
in in lethal radiation studies, they had 72% survival with the Muse cell treatments versus 0 on the control. And then additionally, there's cardiac studies that have a 25% improvement. This stroke study was really interesting from a longevity standpoint because 24% of the patients actually had their gray hair reversed, in
Mm-hmm.
addition to the benefits of having a 32% improvement rate. Um, and then this study in ALS is incredible as well, where it didn't reverse the ALS, but it halted further progression of the disease in 60% of the recipients. So what you could do is get regular, treatments of this Muse and just not progress further in ALS.
Could you go back, please, the slide, the previous one? So this one, what kind of stem cell was it? The one in the middle?
The one in the middle, is what people think of when I went off. I got stem cells today. So the Muse cell is just this brand new cell on the market. It's only being manufactured in 3 labs in the whole world right now, and most of the
So
accessibility.
this is the Muse cell, the one in the middle.
No, the one in the middle is not the Muse cell. The one in the middle is the expanded umbilical cord mesenchymal stem cells,
But
like
what's the
alone.
difference? It also comes from the umbilical cord.
They've also come from the umbilical cord. So
So the
they both
bones.
come from the umbilical cord. And what you do to get the Muse cell, here, I'll go back a little bit. So the Muse cell is in the umbilical cord. The Muse cell is like 1% of all mesenchymal stem cell populations. So there's some Muse in bone marrow and fat as well, at a less percentage than umbilical cord. But what the Muse cell really is, this naturally pluripotent stem cell with superior therapeutics properties. and it expresses HLA-G at 90%, which means it doesn't get rejected, and it can actually differentiate into all three germ layers, and then become directly integrate into tissue. This we call it the Muse cell because it was really discovered and named by this one scientist in Japan who deserves incredible amount of credit, Mari Dezawa, and she was funded by Fuji Medical and was able to do 12 clinical trials on the Muse cell, and it stands for multi-lineage differentiating stress enduring cells. And what she found by accident is that when you have a population of the mesenchymal stem cells, you put them under great stress, the cells that survive are these Muse cells. So you'll make a big population of the umbilical cord mesenchymal stem cells, you'll stress them with acid basically, and 99% of them will die. And that 1% that's left are these potent Muse cell that express this SSAA3, CD105, and then HLA-G at 90%. And then you grow up that population. So the Muse cell is expensive to make because you basically have to do it twice. You have to make a regular population, destroy it, and then make it like a different population of cells. Um,
That's
but
interesting.
at the end of the day, they're all coming from this early embryonic development. You know, these are day 7.5 to 12, where the umbilical cord and placenta are developed, and these are what we can collect at the time of birth without any ethical issues.
But
Um,
when you said like then that you need to stress like the population, how does it work?
Um, the way that it worked, when she writes about it in her book, that she was rushing out of the lab, and instead of putting the regular media into the culture system, she poured trypsin, and trypsin is an acid that is used to remove, like help release the cells that stick to the plastic. So the definition of a mesenchymal stem cell that came out in 2004, as defined by the International Cell and Gene Therapy Society. That's we wrote this article for their newsletter, the ISCT newsletter, and
Mhm.
basically the international ISCT is one of the major stem cell organizations, and they, at the time around 2004 2003, mesenchymal stem cells were mostly being studied from bone marrow tissue, and Dr. Arnold Kaplan from Case Western University was like the grandfather of MSCs, and he was writing all these papers about MSCs, but there was no definition about what an MSC is and how to define it. So, for instance, when I talked about the umbilical cord stem cells, the hematopoietic stem cells, they're defined by one marker. It's a CD34 and CD45, but really CD34 is how you count a hematopoietic stem cell.
Mhm.
To 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 measure and count a mesenchymal stem cell, which is a different type of stem cell, you actually have to find multiple markers. So it's a CD73, CD105, CD90 positive, and these are all done on flow cytometry, which is a test. It's a expensive machine, and you do it by protein antibodies, and then
Mhm.
you measure them with lasers and beads that are track the antibodies, so you can get an accurate count of the cell markers. And then you're negative for some other markers as well, like negative for CD34 and 45 and HLA-DR. And that defines, and then it also has the criteria where it has to be plastic adherent. That's the definition of a mesenchymal stem cell, because when they were defining it, there was no there's no concise definition in the field, and people couldn't compare results because they were looking at different products on
Yeah.
studies. The
But then,
SWE,
sorry, but then, I mean, you're in this industry, right? So you know a lot about this, but like from someone who doesn't really know as much as you do. So, obviously, like if both of those stem cells are coming from umbilical cord, and then they go to the clinic, and for a regular person, for example, like me, it's just like, oh, yeah, like it's coming from umbilical cord. So then like in my brain, like it doesn't register that it could be not a Muse cell. So how, like do you find out that actually like they give you the Muse cell?
yeah. So what you want to look at whenever you get any stem cell treatment, and that's part of the problem, is that there's a lot of misinformation about the stem cell treatments. Um, but you want to see a certificate of analysis, and what you want to see in that certificate of analysis, first and foremost, anyone who ever gets any stem cell treatment, you want to get them to show you the sterility, bacteriology, mycoplasma, and endotoxin testing.